Today, we will be introducing Luminata and its use in impurity management and control. Luminata was developed with a focus on analytical data management and data sharing across your company. Chemists on different sides of the world can easily compare their experimental results using Luminata, making it easier for process chemists to identify impurities and address the impact of process changes to the formation of impurities in their reactions. The ability to compare reaction processes between research teams at different manufacturing and research sites is a major advantage of Luminata, one that can accelerate method development and impurity management. Shown on screen is an example reaction map for a synthetic process, where the different components of the reaction are displayed in a different color. Reagents in blue, intermediates and products in green, and impurities in yellow. By clicking on the components in this reaction process, we can access the analytical data that has been attached to any stage of the reaction. In this case, we have the LC chromatogram and the mass spectra. Reference data can also be added to the process as well. For example, the product in stage 6 of this process has a reference mass spectrum associated with it for comparison. By using the mirror plot option, the chemist can quickly verify that their synthesized compound indeed matches their synthetic target. Next, I will discuss two important data tables used in Luminata: the entity table and the control chart. The entity table stores information about the components of the reaction and is a key data table in the analysis of impurities. In this table, you can see general chemical information about the components present in our mixture, and perhaps most importantly, chromatographic data, including peak areas and retention times. Peak areas are also used in the control chart to calculate the relative concentrations of the different components in our mixture. Using the control chart, chemists can track the formation of synthetic targets, the depletion of starting materials, and the creation and life cycle of reaction impurities. Being able to quickly visualize and quantify these impurities is essential to streamlining impurity control and management for the process chemist. The entity table is linked to experimental data in Luminata. By clicking on the different entities in the entity table, Luminata will indicate to the user which peak in the chromatogram is associated with this entity, highlighted by these blue triangles. Clicking on the different blue triangles will highlight a different entity in the entity table. The mass spectrum panel is also updated when we click on an entity in the entity table. Luminata provides the user with a high degree of data interactivity, all aimed at streamlining data processing and data visualization. Now, I will show how to add a reaction scheme to Luminata and how to attach processed experimental data to the new record in our database. First, we need to navigate to ChemSketch, which is ACDLabs' structure drawing program. Here, we have a synthetic route drawn out with our reagents and intermediates labeled with chemical identifiers. You can also see that we have solvents and catalysts, which are also picked up automatically by the software when we push the reaction over to Luminata, which we will do now. The software automatically identifies common intermediates and merges them. The software is now creating a new record in our database in Luminata. So now we will need to label this process, which we'll do now. Next, we will attach processed experimental data to stage 2 of this reaction. To do this, we first need to switch over to Spectris Processor, which is ACDLabs' analytical data processing platform. Analytical data, in this case LCMS data, was processed and labeled in Spectra's processor, then saved. There are two ways to attach a dataset to a record in Luminata, and today we will do so through the Luminata interface. Let's return to Luminata and attach this dataset to the appropriate reaction stage. By right-clicking on the component of stage 2, we can access the Attach Data option. We will attach the dataset that we just looked at. Upon data import, the software automatically identifies the results of our analysis in Spectra's processor. We have the option to include or exclude components identified during the processing step. Here, we will include every component. When we select the dataset, the software examines the data and extracts information, including the components identified in our processing, along with retention times and peak areas. These data are then used by Luminata to populate the tables we looked at previously, including the entity table and the control chart. You can see that the software has automatically identified the orange-colored components as impurities. Our entity table has been populated, and so has our control chart. The contents of these tables and the associated chromatograms and mass spectra can now be viewed by any member of your company that has permission to access the database. Next, we are going to examine a use case of impurity management, comparing different processes applied to the same synthetic route. You can see here that we have 3 processes with a different wash applied in each case. Luminata has a processing mode called Impurity Master Mode, which allows us to evaluate the types and quantities of impurities created by different processes. We can also identify which process led to the greatest amount of the desired product. Using Impurity Master Mode, we can quickly make assessments about which process was the most efficient. To switch to Impurity Master Mode, we need to highlight the control chart and then right-click on the control chart to select Switch to Impurity Master Mode. When switching to Impurity Master Mode, we need to specify the processes that we wish to compare. Here, as I mentioned previously, we are comparing an acid wash, a base wash, and a no-wash process. We also need to indicate which components we're comparing, in this case, impurities and intermediates, and which stage of the reaction we wish to look at. Upon switching to Impurity Master Mode, we can see that the control chart has changed. The numerical values presented here are the relative concentrations of the individual components in each process. The base wash gave 3 impurities, the acid wash gave 4 impurities, and the no wash gave 5 impurities. We also note that the base wash had the highest overall yield, indicating that this was the most efficient process. If this drug substance is selected for scale-up, the process chemist can now quickly identify which process is best and which one to use in the scale-up. We can also easily visualize variations in the experimental data between the different processes by overlaying the chromatograms in the chromatogram panel. We can do this by turning on chromatogram overlay mode and selecting the different treatments we wish to compare. We do so by right-clicking in the chromatogram panel and clicking Start Chromatogram Overlay Mode. You'll see some options open in the control chart, and by clicking these buttons here, we can select the chromatograms we wish to compare. By zooming in on the chromatogram, we can evaluate differences between the runs. Finally, Luminata can quickly perform impurity carryover calculations at the click of a button. Let's return to our complete reaction scheme and follow the carryover for a reaction impurity. First, we need to switch to reactions table view. Here we can see that a component in stage 3 of the reaction was also present in stage 2 of the reaction. So let's calculate the impurity carryover for this component. We right-click on the component in the reaction scheme, navigate to calculate carryover of impurity, and select it. So you can see here that going from stage 1 to stage 2, about 13% of this impurity was carried over. And then going from stage 2 to stage 3, about 62% of the remaining impurity was carried over to stage 3. The carryover calculation tool simplifies impurity tracking and can help the chemist identify which stages in a reaction scheme are the most troublesome. That concludes our presentation today regarding impurity control and process management using Luminata. This is but a small snapshot of the full capabilities of Luminata. Please reach out to one of ACDLabs representatives for more information about Luminata. Thank you very much.